Recording in progress. Fatuma Shafi.
Oh,
Hello, Shafi.
okay. Sema.
Are you okay? Uko
Yes,
sawa?
niko sawa.
Mbona sauti imebadilika?
Kwa nini? Kwani
Oh,
sauti yangu
inakaa
imebadilika?
si ni juu mnanichezea akili.
Hapana.
Walai hata Gaza ananitext hapa inbox anasema huyo si Shafi.
Achana na Spiderman. Kuna mashida ya masikio.
Umekuwa aje?
Poa.
Nice. Happy to hear from you, Shafi.
Thank you.
Sawa.
Nani alikuwa ana lead cognosy?
Goes.
Niambie.
Leo ilikuwa cognos ama?
Yeah. Nani Hiral is starting now. Hiral is starting in a few. Ama okay, since I don't know. Yeah, share screen.
There.
You guys can see my screen, right?
Clearly.
Kinda.
Okay, so we'll start with essential oils today. So for essential oils, they're like common like properties of essential oils. Like for example, there are many. So the first one is solubility, which they're very oils are not soluble in water, or rather slightly soluble in water, and they're miscible with absolute alcohol, such as rose oil. And then the second one is optical rotation, which we've seen with many other biological compounds, that it's either dextro or levo. So for example, methanol, natural methanol is levo, which minus is levo, the natural one, but the synthetic is dextro. And then there is the various like lemon oil and peppermint oil. Then the second one is color. Most of them are colorless, but when they're left outside and they're not closed, they oxidize, so they get a darker color. Specific gravity mostly is between 0.8 to 1.17. So we know that specific gravity of so they're lighter than water. So that's the sorry,
could you, what's the specific gravity of water?
Sorry, Willie, I can't hear you.
I'm saying
Mhm.
What's the specific gravity of water?
I think it's 1. Let me just check that. Just a minute. Let me just check that. I think it's 1 if I'm not, or is it? Let me just check.
According to Google, it's 1.
Yeah. Yeah, so 1.
Oh, thanks.
Okay. Just 1 minute. Give me 1 minute. I'm sharing again. Just 1 minute.
Yes, I think
Unasemaje Stano?
tunaweza fanya hiyo ama?
I was also thinking the same.
I think kuna mtu anajaribu kuongea bata.
So I shared, or what's going on?
Yeah, I think you keep going, Amma.
Okay, you guys can see my screen, right?
Yes, we can.
Okay. Yeah, so like I said, solubility is one of the properties where they're mostly soluble in absolute alcohol. Optical rotation either have dextro or levo. And then color, they're mostly colorless, but when exposed to air, they oxidize and they get a darker color. Specific gravity is usually between 0.8 to 1.17, and they're lighter than water. So those are the four physiochemical properties of essential
Yeah.
oil. Yeah.
Here in that second statement after solubility, it's saying essential oils rich in oxygenated constituents are more readily soluble. So I was wondering when we sorry, I've muted myself. So I was wondering when we say this statement, like generally, are we saying that essential oils are already oxygenated before being exposed to air or
Yeah.
or some?
Yeah. Yeah, so the four physiochemical properties: solubility, optical rotation, color, and specific gravity. Then, oh yeah, sorry, those are not the only four. Then there is refractive index, then and acid number, which indicates the hydrolysis during the storage process. Yeah. And then one more thing about essential oils is that they don't, like when you put it on like a filter paper or just any kind of paper, it will not leave any stain on it. Yeah. Yeah, and then when you want to, if there's like a mixture of essential oils, you can separate them or identify them individually using thin paper, thin layer chromatography, or gas chromatography. Yeah.
Because like we've jumped into oils out of nowhere. We were suggesting if we could do the crude drugs plant physiology so that it will be an easier flow, because we've moved from, like we've started oils without even covering the basics. Like, yeah, would you be okay with that?
Yeah.
You had prepared for today?
No, I had not, like, I just thought that, because I thought we're doing like alkaloids, yeah, like individually, yeah, but it's okay.
Also we go through plant physiology and crude drugs.
Yeah, that's okay.
Okay. Let me share my screen. I'm assuming you can all see my screen.
Yep.
Yeah.
Great. So today, this evening, we are going to go through just plant physiology, and then we cover crude drugs. Plant physiology and crude drugs. Don't take much time. Right now it's 7:45. By 8:30, hopefully we should be done. We'll start with plant physiology, anatomy and physiology. So plant anatomy. Here, we all know what plant anatomy is, right? We have the things, we are looking at the structure of the plants, the root, the stem, the leaf, what else is there? Just those: root, stem, leaf, flower, yeah, all of those. So that's what we will be focusing on when we're looking at plant anatomy. Then when we go to plant physiology, we'll be looking at functions of various, majorly under plant physiology, we'll be majorly looking at the hormones, the plant hormones, which hormone promotes ripening, which hormone is for this, which hormone is for that, just that. Yeah. We won't go into much details on specific tissues and all of that, right? Then organization, here we are seeing the organization of plants, which is the same as animals or even as humans. We start at the cell. Then when a number of cells come together, they form a tissue. Then from the tissue, we have an organ. Then from the organ, we have an organ system. And at the very end, we have an organism, or in this case, a plant. So it's the same. Remember we said plants are eukaryotic organisms. Plants are eukaryotic organisms. So they have the same as humans. They have the same steps as humans, from cells to tissues to organs to organ systems to the entire organism. So here's where we're going to spend a bit of time. Here, the types of cells. Once we are able to know all of this, we'll be home and dry. So all of these types of cells, epidermal cells, from the basic understanding of the word epidermis, epicardium, right? Epi something. Whenever you see this word epi, you always think of it as there's an outer. This is the outer covering, right? The skin, epidermis, right? So epidermis here functions majorly to do protection, like any other epidermal covering. This one is majorly to protect. It covers the whole, the leaves, the stems. There is where our epidermis is. Then secondly, we'll look at them individually, but we're just mentioning so that once we mention them again, they are familiar. So secondly, we have our parenchyma tissue. And our parenchyma, we'll mention that parenchyma is, this is the most abundant. This is the most abundant tissue, right? Parenchyma. And it's more of a what? It has different functions. It can function from storage and all the way to providing support. All you need to know is that parenchyma, it will be characterized by large vacuoles or spaces, right? Then parenchyma, air. Here is air. Here is just air exchange, gas exchange, air, parenchyma. Chlorenchyma. When you see chlor, this reminds you of chlorophyll, right? Chlorenchyma, chlorophyll. So where so here you'll say it has to do with facilitation of photosynthesis. You know, chlorophyll, green part, the green coloring part that, you know, photosynthesis wouldn't happen without chlorophyll. Then we go next to collenchyma. From collenchyma, I want you to remember collagen. Collenchyma, collagen. What does collagen do in our bodies? It's to offer support, structure and support. Remember, collagen is an abundant connective tissue. So collenchyma support majorly. Then sclerenchyma here, it plays two roles. Number one, it plays the role of support, and number two, transport of water and nutrients. So sclerenchyma doubles as collenchyma, and also number two, transport function. Then we have tracheids. Tracheids with xylem vessels, they go together, transport of water, hormones. Then sieve tube and companion cells, we'll assume they also go together. Here we are talking about transport of sugars, right? So for companion cells, when you hear companion cells, think of the phloem. Remember, xylem and phloem, they go together. Xylem and phloem. Xylem was for water, phloem was for food, right? So now let's look at those basic units one by one. So parenchyma, what have we said about parenchyma? It's characterized by these large vacuoles. Yeah, and it's the most versatile and are common in ground tissue. We look at ground tissue when we're talking about ground tissue, but right now let's just appreciate that it's characterized by large vacuoles. These large vacuoles will be functioning in storage of food items, food products, or just storage of energy, right, in form of food. Here we see a common such as, it's common everywhere. Parenchyma, you will find it nearly everywhere. That's why I say the most versatile. Then the other thing about parenchyma cells is that they can become meristematic at maturity. Meristematic, in plants, just think of meristematic as ability to grow, right? Just think of it as an ability to grow. Meristematic stem cell, meristematic roots, all of that. Ability to grow. Think of it that way. So we are seeing parenchyma cells will function in all of these places. Photosynthesis, but specifically chlorenchyma, gas exchange. Yeah, we've seen gas exchange. It is as aeration, parenchyma, storage, especially storage. Storage is a main function of parenchyma. Storage of starch, protein, fats, oils, water in roots, tubers, and seeds. That's a very major function of parenchyma cells. So take-home message of parenchyma cells, number one, storage. Storage, and it's characterized by large vacuoles. Why are these large vacuoles necessary? To store all of this good stuff, to store the food. The food is found in the parenchyma. So next we go to the collenchyma. Collenchyma, remember we said collenchyma, they are there to support, to provide support and structure. That's the main thing, support and structure. And they are just pectin filled. Pectin, remember it was a type of starch, amylopectin, yeah. But just know they provide support and structure. They'll be found under the epidermis or as the outer layer of cells in young stems and in leaf veins. But the main thing with collenchyma is providing support and structure. They are what makes your plants be able to bend without breaking, right? They also provide that flexibility. Okay. And then for collenchyma cells, we're seeing something unique here, that you won't find them in monocots. Remember, monocots and dicots. We said monocots and dicots are under which division? The angiosperms or angiospermata or whatever. Angiosperms. So monocots and dicots. I hope you remember the differences between monocots and dicots. Then sclerenchyma cells, here we said it doubles. Number one, as providing strength and support to the plant. And number two, transport of water and nutrients. It has two roles: strength and support to the plant, and transporting water and nutrients in the stem. So it has two roles: the strength and support, and number two, also transport water and nutrients. Epidermal cells, we already said epidermal, majorly therefore covering the outer layer, the just in the outer layer for protection. Then looking at the vascular cells, we say here is the xylem and the phloem. The xylem and the phloem. Remember when we said, when you think of xylem, also the tracheids come there. Then when you think of phloem, sieve tubes and companion cells. Xylem, already, just to repeat, we already said, xylem majorly it functions to transport water. Phloem, transport sugar. Xylem is majorly water. This one is, phloem is majorly sugar. Yeah. So these are the three basic types of plant tissue. We have the parenchyma, right? The parenchyma. Here we say the major thing here is storage of food, characterized by the large vacuoles, right? Then we have here collenchyma. Collenchyma, we said structure and support. Sclerenchyma, we said structure and support, and also transport of water and nutrients. These are the three basic types of plant tissue that you need to recall: parenchyma, collenchyma, and sclerenchyma. Yeah. Then let's look at the ground tissue. Ground tissue, it consists of those three types of important tissue types: parenchyma, collenchyma, sclerenchyma, right? Those three types. The ground tissue is what makes up the bulk of the plants. It's what makes up the bulk of the plants. It's there in the stems, in the roots, and in the leaves. Vascular tissue, we've already mentioned. Vascular, we have the xylem and the phloem. Xylem, water, phloem, sugars, right? The epidermal tissue, here in the epidermal tissue, we are having the cell type there is a parenchyma. Again, it's a parenchyma under the epidermal tissue. We said it's protection of plants, the protection, because it's there in the outer layer of stems, roots, and leaves. It's there in the outer layer of stems, roots, and leaves. Finally, we have the meristematic tissue. Remember what we said about meristematic tissue? Just it has the ability to grow, right? So we already said it's under the parenchyma, and it will be on the tip of the shoot, tip of the root, wherever you are likely to find growth, right? In a bud, in a ring around the stem, in woody plants, yeah. So meristematic tissue, just think of meristematic tissue as in our body, the hematopoietic stem cells. It can grow, right? It's not a differentiated cell. It has that thing we call totipotentiality, ability to grow into different stuff. Yeah. The next we're going to cover. So all of this we've mentioned: xylem, phloem. Then we have the cambium, because it's here. The cambium is unique because the cambium is what gives a plant the ability for secondary growth. Secondary growth is whereby you've trimmed somewhere, but the plant is able to grow even after that, right? It's, you know, in maize, if you cut down maize and just remain with the root will stay there and it will not grow. But in some other plants, like even some flowers, you cut them, but they still grow, right? So cambium is that what gives the plant the characteristic of secondary growth. Primary growth is what will normally happen. Secondary growth is after the plant has maybe undergone some damage or something of the sort. So then we're going to look at the leaf. So that summarizes it regarding our tissues. So take home message is this table here. Take home message, this table. Parenchyma, we said parenchyma, storage, right? Characterized by large vacuoles. It's also informs the meristematic tissue. What's the meristematic tissue? It has the ability to produce new growth, tip of the shoot, tip of the root, such stuff. Collenchyma, we said this one is for support, right? Support. Sclerenchyma, transport and also support. Transport of water and nutrients, but also support. Those are the three most important tissues, and that will be the take-home message from plant tissues. Xylem and phloem, that one, we've been covering xylem and phloem ever since primary. Xylem water, phloem, sugars. Xylem water, phloem, sugars. Yeah. Now let's look at the leaf, because the leaf is a site of photosynthesis. The leaf is where we will obtain our food from, or rather, the leaf is where the food we eat will start from. So photosynthesis is worth mentioning. So first, let's just look at the leaf. Because the leaf is divided into two portions. There's the lower epidermis, and now this one we call it the upper epidermis. The lower epidermis and the upper epidermis. Now we are more interested in the upper epidermis. The upper epidermis at the very top of the leaf, or at the, you can say at the surface of the leaf. Sometimes you find some waxy substance, right? That waxy substance. So here let me see here. They've talked about. Okay, they've not talked about it. Okay, you'll find this waxy substance, which is there to prevent unnecessary water loss. Then below that, we have these palisade mesophyll cells. Now, I just want you to know two types of cells when it comes to the leaf: the palisade mesophyll cells and the spongy mesophyll cells. Palisade mesophyll and spongy mesophyll. Palisade mesophyll cells, this is where photosynthesis takes place. This is where photosynthesis will be taking place, whereby you are taking light and converting it to energy in form of ATP. This is where it will take place. But in the spongy mesophyll cells, this is where exchange of gas will take place. That's why you're seeing the stomata here. This is where exchange of gas is taking place, and you can see the spongy mesophyll layer here. It is characterized by these large spaces because there is gaseous exchange taking place here. So we have the palisade mesophyll cells and the spongy mesophyll cells. Palisade, photosynthesis is there. Spongy mesophyll cells, gaseous exchange is there. You can see the stomata there. Yeah. This is what I wanted to call the cuticle, which protects it from leaf, bacteria, and fungi. Then the stomata here, stomata is made up of what controls the opening and the closing of the stomata is this stuff called the guard cells. The guard cells. These are what will control the closing and opening of the stomata. Remember that water loss through the stomata, we're calling it transpiration. So you can say guard cells play a huge role in transpiration. We already said photosynthesis in the palisade mesophyll cells, gaseous exchange, spongy mesophyll cells. Yeah. So that's the take-home message from this, from the structure of the leaf. The palisade, photosynthesis, spongy mesophyll, gaseous exchange. Then the difference between a dicot and monocot stem, we had covered this. The dicot, we said in the dicot stem, we'll see it being the vascular bundle. The xylem and the phloem, we see them, they are in a ring-shaped or in a circular shape. But in the monocot, it's in a scattered fashion. You're seeing it's all scattered. But you're seeing in a, you can see this is in a dicot stem. Again, stem. There's a ring shape here, but in the monocot, we are seeing it's all scattered. Yeah. This one, we want to go deep into this. Then the root. The root, again, we had mentioned this before. The monocot root. Now the monocot root, the xylem and the phloem are in a circular manner, but in the dicot root, they are in an X-shaped manner. Because this is a monocot root. Here we are having the xylem and phloem, they're in a circular manner. Even the pith, you can be able to see the pith. But here it is. In the nini, it's star-shaped or X-shaped, depends on how you look at it, of the root. So the arrangement of the xylem and phloem. For the flowers, we will cover the flowers, ovulation. So we'll go next and just cover the hormones of plants. Then we'll consider that sufficient introduction to plants. We can take a 9-minute break to 8:15. If you have any questions or additions, comments, or what we've covered so far. The plant tissues, take home there, parenchyma, sclerenchyma, and collenchyma. Then the leaf, we've seen especially 2 types of cells, the palisade mesophyll, where photosynthesis will take place, and the spongy mesophyll, where gas exchange will take place. That's what we've covered so far. So any addition on that, you can bring it up. 8:15, we start on plant physiology. Yeah. We're back. Yeah. So plant physiology. Before we go to plant physiology, let's just do what we've covered. We started by looking at plant anatomy. We talked about the different parts of a plant, the root, the stem, the flowers, the leaves. Went into mention the organization, that it is the same for humans as it is for plants. We start at the cell, we go to the tissue, we go to the organ, we go to the organ system, and we finish at the organism. Where in plants, it will be entire plants. In humans, it will be the entire human. Then when we've gone to the tissues, we've seen there are 3 major types of tissue. And these are the same tissues that will form the ground tissue. So the parenchyma, the collenchyma, and the sclerenchyma. The parenchyma, we saw it functions majorly to store, number 1, storage of food, storage of sugars, storage of oils, right? This is parenchyma is where the storage is. And number 2, something unique about the parenchyma, we also saw the meristematic, it's meristematic in nature, right? Meaning it can grow into other cells. It will be found in the root tip and in the shoot tip, where it can grow further. The other thing we mentioned is that parenchyma cells is where parenchyma tissue, the epidermal cells also, they fall under the parenchyma tissue. So the epidermal cells, they function to protection, covering, right, from water loss, unnecessary water loss, and also from infection by the likes of bacteria, fungi. Yeah. Then we've looked at, secondly, we saw the collenchyma. The collenchyma, we said collenchyma functions majorly in terms of structure and support. For collenchyma, we also said we'll see collenchyma being implicated in, or it's the type of tissue that plays a role in the bending of a plant. Why can a plant bend and it doesn't break? There you'll say it's your collenchyma. Yeah. Thirdly, we mentioned the sclerenchyma. The sclerenchyma, we said, and for the collenchyma, we said this is where we'll find our cellulose and our pectin, right? Then number 3, we went and mentioned the sclerenchyma. Sclerenchyma, we saw 2 functions. Number 1 was transport of water and nutrients. And number 2, we saw it functioning also supporting the collenchyma in terms of support and structure. It also has a role there. So these 3 are what forms the ground tissue. What's the ground tissue? It's what forms the bulk of the plants, the stem, the roots, right? That is your bulk tissue. That's your ground tissue. Then we mentioned the vascular tissue. Vascular, just from the word transport. We have 2 types. We have the xylem and the phloem. The xylem, it's what will transport water. It's what will transport hormones. We're going to see hormones here in plant physiology in a few. Then the phloem is what will transport your sugars, right? It's what will transport your sugars. So for phloem, we mentioned a specific type of cell called companion cells. They are what make up the phloem. For xylem, it's just the tracheids. Xylem, remember, xylem is just a tube. Think of it just as a vessel. But phloem, it's made up of cells. Yeah, it's made up of cells. Finally, we mentioned that arrangement of xylem and phloem. In monocots, in the stem, we say the xylem and phloem, they are scattered all over. But in the roots, they are in a ring-shaped manner. But for dicots in the stem, they are in a ring-shaped manner. In the root, they are in an X-shaped or star-shaped manner, right? So that's what we've covered so far, xylem, phloem, and those types of tissues. So now we go to plant physiology. We looked at the anatomy. Now we look at the physiology. Physiology, we know just from basics. Anatomy deals with structure. Physiology deals with function. And when we are looking at plant physiology, we'll focus on number 1, the photosynthesis, because that's the most important part to humans, to even other animals, photosynthesis. And then we look at the hormones, the plant hormones. We'll just mention briefly the tropisms and the nastic movements, okay? So let's start with photosynthesis. Photosynthesis, we know what we say about photosynthesis. It's simply a process whereby the plant will utilize that light to make ATP, to make food essential. It's that process. Plant will utilize ATP to make food, right? So photosynthesis, where does it occur? In the chloroplast. The chloroplast. Remember, we asked even mentioning the chlorenchyma. It occurs in the chloroplast. And we'll see the chloroplast. There are two types. It consists of two areas: the grana and the stroma. The grana and the stroma. Now, for photosynthesis also has two reactions. There's a light reaction or light dependent reaction, and the dark reaction. The dark reaction is also called the Calvin cycle. So in photosynthesis, now we've said in the chloroplast, there's a stroma and there's a grana. So the light dependent reaction, it will take place in the grana. LG, life's good. So light grana, right? You can catch it like that. LG. So light dependent reaction will take place in the grana. The grana is where you will find our chlorophyll. This is where we'll utilize light from the sun to form our ATP. We're forming here our ATP. Then our ATP and also NADPH, we'll throw it in the Calvin's cycle. We'll throw it in the Calvin cycle, and we'll see what the Calvin cycle does. Just in a summary, not so deeply. Yeah. So chloroplast consists of the grana and the stroma. Where does the light dependent reaction takes place? In the grana. So the dark takes place in the stroma. Dark also, it's also known as the Calvin's cycle. So yeah, we've already said light energy will be captured in the grana and packaged as ATP and NADPH. Then the sugars is now what will be formed in the Calvin cycle. In the just from this depiction, from this diagram, in the light dependent reaction, we'll just be forming ATP and NADPH. Then from Calvin cycle is where we'll be forming our sugars. It's where we'll be forming our starch, all those stuff. Yeah. So here it is. So we already mentioned this. We're not going to go deep into how that happens. So, but here now we have the Calvin's cycle. We are seeing something just to mention in the Calvin cycle. You're seeing Calvin cycle requires CO2. This is why we say plants do take up CO2 from the environment, because they require it to form your sugar. They require it to form those starch, those stuff. Yeah. So that's why plants take up CO2, they'll release oxygen. Yeah. So here it will take up CO2. We're not going to look at all of these steps. All we're going to mention is that once it has taken up CO2 through a series of steps, we are seeing here, we will form what? Our sucrose and other sugars, including starch. Yeah. So we form our sugars in Calvin cycle or in the dark reaction. So that is it. That is it with photosynthesis. Just know where it takes place in the chloroplast, and that there are two reactions that takes place: the light dependent in the grana, right? Life's good, light's grana. Then the dark reaction takes place in the stroma, also called the Calvin cycle. And the stroma is where we will have our sugars being produced, the likes of starch, the likes of sucrose. Okay. Yeah. Then finally, we can go to plant hormones. So plants, like humans, they also have hormones. Plants are complex organisms. They're complex organisms. They're not as basic as bacteria. So they also have various hormones that help with different defined roles. And these hormones, these are the common hormones: the auxin, the gibberellin, cytokinins, ethylene, and abscisic acid. These are the common ones: the auxin, the gibberellin, cytokinins, ethylene, and abscisic acid. Now, I just want to display where the function of these hormones are listed. A second. Yeah, okay. So let me mention the functions and then we look at them again. So auxins, gibberellin, cytokinins, ethylene, and abscisic acid. Auxins, let's start with the auxins. Auxins, they are what will stimulate plant growth. And by plant growth, in this case, we are talking about the plant growing upwards or downwards. Let me say that in simple terms, upwards or downwards. So the plant, the shoot shooting up, or the root going down. Something unique about auxins, or it's something we do at home in our gardens without realizing it, is that auxins, they favor that upward growth or downward growth. Now, for the sake of our gardening purposes, let's say auxins favor upward growth, and you will find auxins being concentrated there in the shoot, right? So you notice once you trim your shoot or that upper part of your flower at home, that flower starts growing sideways, right? Because auxins, they will stimulate that upward movement, but inhibit, or upward growth, but inhibit lateral growth. They inhibit lateral growth. Yeah. So auxin will stimulate that upward or downward, but inhibit lateral growth. Upward, but inhibit lateral. So next, after mentioning auxins, we'll look at cytokinins, because cytokinins now do the opposite. Cytokinins, they stimulate lateral growth, sideways, going sideways. That is cytokinins. They stimulate lateral growth, but inhibit that upward growth, So if you want your plant to just go up, 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 you want to cut those branches that are going sideways. Cut them, your plant will go up, because cytokinins won't be released to inhibit that upward growth, okay? Yeah. So next we go, so auxins are growth, but upwards, downwards. Cytokinins, lateral growth. So here that from root tips to the base, and for the plant form through cell growth and expansion. Auxins from the apical bud, it will suppress the growth of lateral buds, which sprout when the plant tip is cut. So this is what we've mentioned. You cut the upper tip of your flower at home, it starts growing sideways or lateral. Next, we go to gibberellins. Gibberellins, you just want to remember two things with gibberellins. The first thing is that they cause stem elongation. That's the first thing. They cause stem elongation. And the second thing with gibberellins is that they break seed dormancy and initiate germination. They break seed dormancy. So once you have your seed in the ground, you've planted your seed, what will break that seed dormancy and initiate germination is this gibberellins. It's this gibberellins, right? So here it is, they can be commanded for tall growth. Cytokinins, we already said they promote lateral growth. They promote lateral growth. And also the other thing, they delay aging in plants. They will delay aging. Now, because we've mentioned aging, next we'll go to abscisic acid. We'll finish with ethylene.
Kyle,
Abscisic,
just a minute. You said, just go to the previous slide kindly. You said they delay aging. That's for gibberellins, yes?
No, that's for
cytokinin.
cytokinin. Gibberellins,
Okay.
it's for breaking seed dormancy and internodal or stem elongation. Let's call it stem elongation.
Okay, thank you.
Yeah, cytokinins, that lateral bud development and delay aging. So if cytokinins delay aging, what promotes aging? We have abscisic acid. Abscisic acid, number one, it promotes aging. Number two, abscisic acid will also promote, it can induce dormancy, right? It can induce dormancy, and it's also used to maintain that dormancy. Dormancy, in this case, dormancy is the same as we mentioned when we were talking about spores. It's just a state where this organism, whatever it is, it doesn't require a lot of nutrients, right? Dormancy, it's like, yeah, it's what we mentioned with the spore-forming organisms, the Clostridia and the Bacillus. It's the same here. Dormancy is just a situation where that organism won't need much nutrients. So that tells us abscisic acid also plays a role in detecting what? Stress in the environment. Stress, in the case of a plant, what constitutes stress? It's stuff like there's no water. There's no water in the environment. If there's no water in the environment, what does you can think of abscisic acid as the stress hormone of the plant. If there's no water in the environment, abscisic acid will, number one, promote aging. Number two, can induce dormancy. And number three, you see this, it can, remember we talked about the stomata and we said the guard cells. The guard cells is what is involved in the opening and closing of the stomata. So if there is no water, the abscisic acid stimulates even the guard cells to close up the stomata so that you do not lose water through, so that the plant does not lose water through transpiration. So abscisic acid is your stress hormone. It signals water stress to the plants, and it's produced in the leaves and roots under stress, right? So this is what abscisic acid will control, will prevent water loss. It will be induced in times of stress. Finally, we can look at ethylene. Ethylene is what will promote your fruit to ripen, right? So ethylene will promote, number one, your fruit to ripen. And we know if it ripens so much, it can also rot. So ethylene can promote ripening and even rotting of a fruit or your plant, not just your fruit, or your plant. And number two, ethylene, it will also play a role in pathogen attacks. Remember, ethylene, if we can remember from organic chem, when we're doing organic chem, ethylene will exist as a gas. Here it is. Oh, yeah. Ethylene will exist as a gas. So once, for example, you have two plants next to each other, ethylene will be produced, and because it's a gas, it can diffuse to the other plant, and the other plant will read this signal as, hey, yo, my guy, I'm getting infected here. There's a pathogen attacking me. Stay safe out there. Okay? Stay safe out there. So ethylene was playing a role in that. Pathogen attack. It was playing a role in that. Pathogen attack. It's a gas, so it can diffuse from one plant onto the next plant. Yeah, signaling that everything is not okay. Then finally, there's one that's not mentioned here. It's called jasmonic acid. Jasmonic acid. So jasmonic acid is for wound healing and repair. Wound healing and repair, it's that jasmonic acid. It also plays a role in defense, but it plays a huge role in wound healing and repair. That's jasmonic acid. Yeah. So next we go to so for plant hormones. Let's go here and just mention. In a blank page. Yeah. So for plant hormones, we've mentioned 5, or rather we've mentioned, let's say 6 hormones. 6 hormones, right? We started with auxins. Auxins, we said their main role is that upward growth. Let's just say upward growth. And they also inhibit lateral growth. Growth. Yeah. So that was auxins. This was the main function of auxins. Secondly, we mentioned the opposite of auxins was the cytokinins, right? Cytokinins, now they were promoting lateral growth. And number 2, we saw that they delay aging. So number 3, because we've mentioned aging, which one also has a role in aging? Abscisic acid. I don't know abscisic acid. I don't know the spelling of this thing. Wherever just write it like that, but that's wrong. Abscisic, whatever. We say this one, it promotes aging. Number 2, signals water stress. So has a role with stomata closure to avoid water loss. Then the other hormone we mentioned now was the gibberellins. What did we say about gibberellins? Stem elongation, and number 2, most important, breaking, seed dormancy breaking. So this is what will induce germination. You can say it induces germination. Those are 1, 2, 3, 4. Next, we look at now the ethylene. Ethylene, number 1, we said ripening. Just say ripening, and also this one, they induce aging. Remember, the other word of aging is senescence. They induce aging, but also role in pathogen attacks. Right? It can diffuse from one plant to the other. Yeah, to signal the attack. Finally, we finished with jasmonic acid. Jasmonic acid is so number 1, wound healing and repair, but also have a role in pathogen attack, production of toxin, pathogen attack. Yeah. So this is a summary of the hormones, because the hormones are the most important ones to remember when it comes to plant physiology. So this is a summary of what those hormones do. Yeah. Auxins, upward but inhibit lateral. Cytokinins, they promote lateral delay aging. So this is the principle of pruning, right? Abscisic acid, we saw they promote aging, they signal water stress. So in that regard also, we'll see here, it will maintain dormancy and such stuff. Gibberellins, stem elongation, but breaking seed dormancy. So germination. If germination is not happening, we want to look at gibberellins. Are there enough gibberellins? Ethylene, we already said ethylene, ripening of the fruits, and of the plant in general, but also have a role in pathogen attacks because they can diffuse from one plant to the other, signaling that there's an attack going on. Jasmonic acid, wound healing and repair, and also in the pathogen attack. Yeah. Unless someone has something to add in terms of physiology, that will be it. Just from photosynthesis, remember light reaction, dark reaction. Light takes place in the grana of the granum. The dark in the stroma. In the dark, that is where we call it Calvin cycle, and it's where we'll produce our sugars, our sucrose, our starch. There is where we'll produce, and there is where we'll utilize CO2, right? There is where we'll utilize CO2 to produce our sugars. In the light reaction, we just produce ATP and NADPH. They take place in the chloroplast. You can say chloroplast falls even under the parenchyma. Okay, all of that. Chlorophyll, green coloring. Yeah, don't need to mention that. So if anyone has any addition, you can add it now. While we take a 4-minute break, because crude drug is just a walk in the park. Will be done in 10 minutes. So 8:45 we start crude drugs. 8:55 we finish crude drugs. Yeah. So, just to reply to a message, what's the difference between stem elongation and upward growth? The difference between stem elongation and upward growth. Allow me to share just something on the anatomy of a plant. It will, and also highlight here something. Stem elongation was just a simple term to say that what it actually does is causes stem elongation between nodes on the stem, or it causes internodal, it promotes internodal growth. So the next question is, what are the nodes? The nodes is the parts where we are seeing from one leaf to another leaf, from one branch to another branch. Those are the nodes. Let's go there so that we just see that. Yeah, it is. See, we call this is one node, this is one other node. So what gibberellins will do, they promote internodal elongation, the distance between this node and this node. That is how they call stem elongation. That is how it's different from the upward growth. Auxins will be working here. This is where auxins will be. This is where the auxins will be highly concentrated. That's why when you cut this off, this plant will start growing lateral. That will be growing lateral. So auxins are here for upward growth. Yeah. But now the gibberellins will be, you can think of it as they'll be working in this portion between the nodes, promoting this length. If it's 5 centimeters, If I spray these plants with gibberellins for a good time, the next time I check, it can be 7 centimeters. That internodal stem elongation. Yeah, it's a very good question. Internodal stem elongation. Hope I've answered your question.
Wait, so you're saying stem elongation, the nodes have to be involved, and then upward growth, it's the top part.
Yeah, exactly. Yeah.
So thanks.
Oh, I'm seeing even there's something we forgot to mention. Okay, we're going to cover that, then cover now crude drugs. But that one is quick. So you guys remember tropisms. Tropisms we're saying how plants react to external stimuli, right? Yeah, and we're having phototropism, geotropism, hydrotropism. Yeah, all of those types of tropisms. So for tropisms, I know it's just an easy one. Tropism is simply, we are talking about how plants react to various external stimuli. So phototropism, remember that experiment whereby you place your plant, you cover your plant with a container, then you drill a hole somewhere where the light will be coming from, and the plant will grow towards that hole. That's phototropism. The plant will go looking for the light. It will follow the light, right? Then hydrotropism, hydro means water. Here, majorly, we'll be looking at the roots. The roots will go far and wide looking for water. Gravity is the pull of objects to the center of the earth. The roots always grow downwards to the center of the earth. And here, the influencing factors, we'll see the influencing factors are the auxins, indole acetic acid. These are the auxins. IAA is another term for auxins. Why auxins? Let's look at phototropism. The plant will be growing towards the light, looking towards the light. What grows towards the light? The shoot. So and we know the auxins will work on the shoot for the plant to grow towards the light. Then looking at geotropisms, remember I kept saying upward and also downward growth. Upward for the shoot, downward for the root. The auxins work in those two places, in the shoot and in the roots. So in the root, again, geotropism to promote it to grow towards the center of gravity. Hydrotropism is now towards water. We have another called chemotropism. Chemotropism is now towards certain chemicals, things like fertilizer, maybe phosphate, depending on the plant. Then inergistic movements. Remember we said plants are immobile, right? Plants can't move from A to B, but they can respond to touch, can respond to wind, vibration. Simply, inergistic movements is whereby we see plant parts. Plant parts will react to an external stimuli. For example, there's these plants, you will touch the leaf and it will fold. So that's what we call a inergistic movement. Yeah, the plant part moves, but the plant itself is stationary. We know the plant can't move. Yeah. So finally, let's look at crude drugs. Let's look at crude drugs. Yeah, so crude drugs. Crude drugs, essentially, it's just what the take-home message from here, from crude drugs. I just want you to think of the drug in its natural form. Of course, when we are talking about crude drug, we are talking about drugs that are obtained naturally, right? And in this case, majorly we are looking at the plants. So when we are talking at crude drugs, we are saying a drug in its natural form. For example, something like the quinine. We know quinine is obtained from the bark of the Cinchona species, right? So if I take that bark and I just cut it into small pieces. That's my, you can think of it as, that's my crude drug, okay? I haven't modified it in any way. I haven't done anything to it per se. It's just here, the crude drugs are plant, animals, or their parts which after collection are subjected only to drying or making them into slices, pieces, or peeling them in some cases. They exist in their natural form. It's something I just like to show you. Yeah. So we know that the poppy plant, the morphine is obtained from this opium fruit or seed. Yeah, this is where we get opium from, right? This is a plant, right? This is the fruit. Okay, most books call it the fruits. These slides call it a seed, but most books call it the fruit. So I'll call it a fruit, right? We know we obtain morphine from here, from this fruit. So let's say you've planted this stuff illegally and you have morphine ready to harvest. Here it is, the fruit is there. So let's see from here, I want us to understand what a crude drug is. So you have your that fruit, then now you just take the juice, this juice that is flowing here. You cut it and you take this juice and you say, put that juice in a glass. That will be it. That will be your crude drug. You haven't done much, right? You haven't added any chemical. You haven't, literally, you have not done anything. The drug is still in its natural form. Or if you, to put it better, the active ingredient has not been extracted. Because we know inside, in this fruit or in this juice, it's more than morphine that is inside there. It has other stuff. It has other ingredients, other chemicals in it. It's not just pure morphine inside there, okay? So that's what a crude drug is. So a pure drug, we get a pure drug after we've extracted from the crude drug. Now from the juice, we said from the juice, there's morphine, yes, but there's other stuff. So when you specifically extract morphine, then you will obtain your pure drug. And we'll look at the extraction techniques later, the likes of maceration and all that. Yeah. So when you extract, now you have your pure drug. But then now once you have your pure drug, you go and make it into an injection, into tablets, into a liquid. Now there we'll have your pharmaceutical preparation. So your crude drug, just think of your crude drug as it's there in its natural form, right? You haven't really extracted the active ingredient, in this case, morphine, right? If it was a cinchona species, I haven't extracted quinine. I still have a mixture of whatever is there in the back. Yeah. So a crude drug, we already defined that. And crude drugs, they can be classified in a number of ways, from alphabetical, morphological, taxonomical, pharmacological, all of these ways. Alphabetical is simply from A, B, C, D. There's nothing much there to be mentioned. It's this way: acacia, agar, benzoin, whatever. Morphological here, it's now it's according to the part of the plant or animal represented into now organized and unorganized. Here, now I want us to just look at it in simple terms, in that they can be crude drugs in terms of morphological classification. They can be either organized or unorganized, right? They can be organized or unorganized. Now for the organized, here we are talking about we can obtain our drug from a specific plant, a specific part. We can obtain our drug from a specific part of the plant, right? So if it's the quinine, we can obtain quinine specifically from the bark of the plant, right? If it's the digoxin, we can obtain it from the leaf of the digitalis. If it's morphine, we can obtain it from the fruit, right? We will call that that one will be organized. That one will refer to it as organized. But when we're talking about unorganized, here we are not obtaining our drug from any specific part of the plant or the animal, but let's just focus on the plant. You're not obtaining from any specific part per se. You're just like, it's not like this nini is concentrated somewhere in the leaf. It's not like it's concentrated somewhere in the bark, in the root. No, right? You can see here in organized drug or cellular drugs, even from that cellular, meaning they're in certain cells, in the roots, in the leaves, in the fruits, wherever, right? But in unorganized, we are seeing here the examples. It's not from any specific part per se. It's not from any specific part. It's just that, hence that term, unorganized. We are not obtaining our drug from a specific part. So in short, here the classification is based on the part of the plant where we're obtaining the drug. If we're obtaining it from a specific part, then it will be organized. If it is from no specific part, it will be unorganized. Yeah. Then chemical classification is where we have the alkaloids, the glycosides, tannins, all of those. Yeah, phenols, volatile oils, whatever. That one is based on the chemical structure, right? And we know this is the most common classification, right? The alkaloids, the glycosides. Yeah. Taxonomy is just based on which family it's obtained from, which family, which genus, which species, all of that. That's taxonomy. The other common type of classification of crude drugs is based on their pharmacological action. It's based on their pharmacological action. For example, we know morphine. Morphine is an analgesic. So it's a crude drug, but we can classify it as an analgesic crude drug. Another type is digoxin. We'd say it's a cardiac drug, or cardiac antitussive. Yeah, it's just based on the pharmacological action, cardiotonics, digitalis, such things. Emetics, we have this chachuna, it's usually a funny name, right? All of that. Cardiac depressant, just based on the pharmacological action. Then, yeah, that is it about crude drugs, honestly. Just crude drugs, take-home message is just what I want this here. It's in its natural form. It hasn't even been extracted. That's all I want you to remember from a crude drug. It's in its natural form. It hasn't been extracted. Yeah. So that's it from me. That's it from me. Unless there is any addition.
I think one of the questions that you guys should focus on, because if you go to the questionnaire, the ones that the PPB ones, you'll see you've been asked to match a crude drug to the origin. So if you can have like a template, for example, like the vinca alkaloid, what it produces, something like that. Like for maybe a summary of all drugs. I had it, but I can't seem to find it. You can share with everyone.
So drug, you said a drug and its source, like vinca alkaloids, and now what
No. Like you see the digitalis, digitalis produces, like this the active drug. What does it produce? You see, like morphine is produced from a
understood you.
Yeah.
Okay. Let me see if I can find a good tag. Nice. Divya, any additions? No, yeah.
There's a list. If I find it, I'll share. Not anesthesia. I don't think you have understood my question, but I'll show you. When I show you, to make sense. And if anyone else can find it, well and good, because for a fact that question will come in the PPB exam. Like, it has always been there.
Yeah, I'm not getting the question. Are you saying like kind of like the way what's it called? Coffee plant produces like morphine, such a thing.
Exactly. Now there's like a class. Or, okay, now for example, if you ask digitalis, what does it produce?
The goxin, I think.
Exactly. Now you see, now those ones. So you'll be
Oh, so
given
now we have to match like the source of the
yes,
drug
yeah,
and the drug it produces.
exactly.
Okay. So like the atropa belladonna used to produce what? I don't know.
It's opium.
Okay. Okay. Unless we search for it on the internet. Let me see if there's such a table on the internet.
But when we actually go through the questions, you'll just see it.
I think we'll also come through that when we're looking at individual, like alkaloids.
Yeah.
Individual.
When we get the classifications based on the chemistry, we'll get to see more of that. For example, now when you go to digoxin, this is part of the cardiac glycosides. So we'll be able to appreciate that aspect, I think, when we go into details.
Oh, I think like even this one is like sort of like it shows, yeah, the one that has been circled in yellow.
But a short list, yeah.
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 Okay, then maybe the more we read on it, and then I can make a table on everything and then summarize them, and then just like post the table with the summary.
Okay.
All right. So tumemaliza for today, ama?
Yeah. Unless mtu ako na something to add, yeah.
Thank you.
Okay.
Thank you for the summary. Alafu guys, see when Patrick makes the calculation for the amount we're supposed to send for the Zoom to tume tu pesa, like, yeah, it's really helping us. So if we can all just sacrifice something small and send the money so that we can get the Zoom credentials, as opposed to like the last time when it took almost more than a week to get it. Yeah, that's all.
Yeah, meanwhile, mnaweza join ya Kabarak sasa, because I think ya Kabarak is going till 10. They are revising paper 1, so if you have energy, you can join that one also. Yeah. The Google Meet, it's the same. If it's new, wametuma kwa the group. Yeah. See you guys. Tomorrow, bright and
Kama
early.
hataki kuona.
Where's Divya? Divya is here.
Yeah,
Divya alishaenda.
See you tomorrow. you are our champion.
For?
I think you're doing folk
Oh, yeah,
medicine. Yeah,
Uh-huh.
yeah.
Okay, let me ask for those. Do we have notes? Because I was just going to use the slideshare because I've not seen where those notes are.
Yeah, just
Which one's
use
Divya?
SlideShare. Just use SlideShare.
Okay. Yes, Stan.
Oh, sorry. I was just asking which notes. The
No,
one
I was just
for guy for
folk
Guinness, Ama?
medicine.
The guy who used to drink Guinness. I've forgotten his name. Ombija.
Oh,
I don't
yeah,
know.
that one, Bingy.
Oh.
Yeah, I
Yeah.
think there are notes.
They are. Okay, I'll
Yeah.
have a look at them and
Okay.
then I'll present. Wait, Hiral, you're not doing oils tomorrow?
I can do oil, by the way. I can do tomorrow because, yeah, I can do it tomorrow.
Okay, just do it tomorrow because I didn't have the time to prepare on this weekend.
Okay, so
That's
I'll
okay.
start
That's
with,
okay.
I was really
start
busy.
with essential
So I'll
oils.
take you guys through the day after. Yeah,
Okay,
that's
fine.
okay. All right, thank you.
Absolutely lovely.
So tomorrow we are doing oils.
Yeah,
Yes.
we can do oils tomorrow.
And also, oils is a very short topic, so we can even do something else. It's not much to cover.
Okay.
Okay, if I'm ready by tomorrow, I'll start. There's
We
no
can
problem.
start with oils and then we see what we can move on to.
Yeah, no problem. That's fine.
Okay, fine.
All right. Good night, guys.
Good night.
Good night. Good night, Divya.
Good night.
Good night, Leting. Guys, kwa me good night. Where is she?
Hata singoje.
No, good night.
Stano,
Shafi.
nilikuwa away for a while. Mlikuwa mna discuss nini? The last bit.
Oh, tumemaliza na crude drugs. Juu ulituacha wapi?
Mkiongea about Kabarak, what's that? We're joining another discussions. Oh, that's fine.
Yeah, Kabarak. Yeah, there's another discussion. I think there's a group that was. I don't know if you joined that group by any chance.
The one that Serafina added me.
No, that one, not that one. Another one. Inaitwa PPB revision.
Banaambia wala wajaanza Kabaraka wajaanza bado.
Sijaona link hata. Hakuna link nimeona umetuma.
Oh, so you can add me. I'll join that one too.
Okay. Are you Shafi? Ama sinaona ni kama uko hapo. Let me see the participants.
Oh, nimeadiwa sahi na Tevo.
Yeah, nimekuona.
Oh, niko hapo. Sawa, thank you.
Shafi. Welcome any day, anytime.
Sawa. Have a good night, Stano, Spider-Man, and everyone. Tevin, Brenda, Shrina, and all. Bye.
Nawambi Constant Chiri.
Constant Shiri, hi.
Yeah.
Oh.
Constant Shiri ni wa wapi Gauss, ni wa Kabarak?
Welcome.
I think so. Anaweza tuambia Constant manze, kwanza Kabarak vile mmeivisha pharmacognosy.
I know.
Umeona hadi hizo notes wewe? Nimeona bana.
Notes unazisoma pharmacognosy inaingia tu.
Tutumie hizo notes. Wako poa basi.
Wewe kwani huko Kabarak wewe Shafi?
Siko imagine.
Wait, Stano, something hizo notes.
Labda connections.
Connections, my guys.
Change hizo, tuma.
Nitatuma. Mzae, kwani unatumia hadi ring light? Mbona sauti inatokea vizuri hivi?
Chorea.
Nice one. Constin ni kama ameenda.
So kesho ile masaa yetu stano asubuhi na mapema.
Ile masaa tu mazee, hiyo
By the way, hiyo
kesho
nitajoin.
Nice, welcome.
Nitumie link ama ni hii tu?
Link ni hii tu.
Link ni the same.
Sawa.
Wewe wenye ni masaa tu unafaa kuwa nayo.
Ni 10 am nimeambiwa.
Masniper. Wow. Kumbe saro masniper hapa.
Aje Tevo?
Mko na link ngapi hivi?
Link ni moja tu.
Naona watu wamechangamka.
Link ni moja, but kuna discussion ya 10 to 12, then kuna sasa usiku, but link ni the same tu.
Top sawa.
So 10 to 12, 10 am to 12 pm tutakuwa tunafanya, then tunaendeleza usiku, because tuliona sasa watu ni the same wenye wanakam discussions, so tuna maximize time.
Top. I'll be there. Ndakuwa hapo.
Top.
Mr. Appreciate the aspect.
Unasikia chai tunaambiwa top. Mbona mimi nikisema top wewe unacheka? Si poa. Hai Mumbi.
Tutaezana sana. Lugha ya cool kids tutaezana.
Top. Wamesema tunaanza na hiyo tu.
Chorea.
Top.
Mr. Appreciate that aspect.
Ni Wakabarak ni kama leo hata nani.
Naambiwa ni kama ilisambaratika kati watu waliamua
group.
Yeah. Like ati group imekuwa kubwa sana, but wanaanza kesho. Like wameform smaller groups but wanaanza kesho. Yeah.
Makes sense. Kila mtu ni kung'ang'ana. Guys, let's call it a day.
Unajua place mtu anaweza fanywa HbA1c, place poa around Thika Road.
Around Thika Road. Guru Nanak ni Thika Road.
But hiyo lazima ulipe consultation, nataka tu msee afanyiwe.
So unaingia tu unaenda lab direct then unalipia unasema umetumwa HbA1c test.
Na ni how much?
I think the last time ilikuwa I think ni 12 so ama ni ngapi hapo around.
Okay, so
Na hii
ukiwa
avenue
na
ya Garden
ngiri niko
City.
fiti. Sasa sijui
but hata hii watatuma kwa main branch. Hebu nione. I don't think wako na lab.
Sitaki hiyo kutuma, nataka ya kupata results within a few.
Yeah, huko unaenda tu direct kwa token machine, unasema unaenda lab, then unaenda kulipa, then unapelekwa lab, unaprintiwa result zako. Si lazima uende kuona doc. Another one pia unaweza enda but I don't know, I think maybe hadi kuangalia invoice pia nani si mbaya. Inaitwaje Lancet. Lancet kwanza ni chap mbaya sana. But I don't think kuna Lancet kuna nayo Thika Road hakuna. Hadi Second Avenue Parklands.
Yeah, I think hadi Parklands.
Hiyo ni Parklands. Kwa kuna msee unashuku nani juu sugars.
Yeah, iko a bit fluctuating. Mara normal, like fasting, mara normal, mara high. Sana nataende sasa.
But nani fasting ndo inafaa fluctuate ama ni random?
Ni fasting. The other I think last week but one ilikuwa fiti kitu kama 5.1 like yeah, but then leo asubuhi inakuja 8.
Ako on meds.
Ako tu nini? Ako ako tu on antihypertensive meds. Constant ni wewe tunangoja uongee by the way ndio tu stop hii meeting.
Mazee tuna constant bana. Constant Moshiri ama naita nani? Constant Shiri. Kama hata alienda hata ametoka.
Amelive. Umembuli.
Tumembuli. Ni kama wewe unanimenya ameingia saa hii huyo dem akatoka hiyo.
Kosi night night.
Kivuti. Kama nito na.
Night night.
Nini 99?
Mresha anajiita night night.
Unajuaje ni mresha?
Siona night night.
Telling text.
Unakadvishiwa mzae.
Ni kama unajua gas ni fala sana. Gas lete mtu lienda online alikuwa anaingia na account fulani sijui ni alikuwa amejiandika Naomi ama and then alikuwa anashinda kini text.
Inaweza kaniki
kifala. Hata hizi tuma pesa.
Unajua tulikuwa na class yetu tulikuwa na boy fulani alikuwa fala sana. So unapata jamaa anamwandikia barua klasi yetu then anasema ni demu wa Moiforces but ni jamaa tu wa klasi yetu tu. Then anaandika namba yake then holiday anamtumia tu ma credit tu airtime, anamtext ni aje silo nitumie airtime ya pao. Jamaa anaenda nunua na buy band.
Stop. Lazima tusim.
Nilishangaa sana. Hiyo ilikuwa top hiyo Tevo.
Ah,
Mm.
Ladies.
kwa hivyo Stano umesema kesho 10.
Yeah, 10 a.m.
Ah,
Same link tu.
safi.
Then after hapo ndo endo ichuma sasa.
Vizuri sana.
Leo umeingia?
Lazima.
Mm.
Ujaingia leo?
Kazi nimeingia, si nimekuwa na huyo beshte yako huko.
Gani hiyo?
Chai.
Ah, kijana mtiaji sana.
Piaji sana. Chai we kijana top.
Mzee unasemaje?
Umeshapiga ile peanut sauce yako pale na matoke?
Piga nutmeg yake. Unajua tunaenda kwa oil.
Nimeingia.
Nutmeg mimi naikanga hadi kwa nyama siku hizi.
Wala.
Inaekanga kwa nyama pia.
Sijai try.
But haiko ile kama potted kama kwa chai direct. Unaweza kwa chai unaika direct unai absorb in the top.
Chai. Nakuanga potent.
Yeah, mama potent sana.
Kukombero.
Wewe uliniambia nikubebe muhombere bana, alafu unaniruka.
Mzee unajua sikuwa najua ati ni nani unabai. Nikafikiria ni yako.
Zizi.
Nishangaa
Piga nutmeg muhombero na cassava na
salt.
Na njugu. Utakuwa unashinda kwa carpenter kila siku. Kuni blender kwa edge,
kijana wa Ipekakuna.
Inaitwa Ipekakuna. Hiyo jina sijaishika.
Niite hivyo.
Ipekakuna ni nini?
Hiyo bibi fulani.
Ya emetin.
Yeah. Ipekakuna.
Ati active ingredient ni nini?
Ni plantain na hapa nini? Hiyo
Emetin.
Yeah. Ya kuinduce vomiting kama mtu amepoisoniwa. Ipekakuna. Chai ananichocha hapa. Nitaenda kusema hivyo mahali nicheke.
wewe ndio ulisema hivyo. Na ndiye alikuwa anasema Ipekakuna.
Alkaloid. Ipekakuna. Mwana Gauss liona nani alipeleka watu World Patient Safety Day, kitu kama hiyo. Hermes.
Aliwapeleka?
Alipeleka kama of the 50 years mchanganyiko tu. Alikuwa na representative wa students niliona. Walikuwa kama
walikuwa huko. Hermes alikuwa
alikuwa. Imagine.
Mimi niliambiwa hakuwa, imagine.
Alikuwa
mimi nimemuona.
Basi alikuja privately.
Hey.
Guys,
So
hebu
ladies,
tutoke, itakuwa refu for no reason.
Gausu, umesemaje? Sijasikia. Oh, pole.
Recording itakuwa refu for no reason, so tunaweza toka tu because it keeps
Acha
recording
tuende tu.
until the last person leaves. Ni automatic, si
By the
manually.
way, itakuwa ndefu sana. Sawa.
Baadaye.